Hi everybody, welcome back to this short course of bioinorganic chemistry. We have been discussing about the metals in medicine. Metal ions play very important roles in biological processes, and the field of knowledge concerned with the application of inorganic chemistry to therapy or diagnosis of diseases is medicinal inorganic chemistry. In my last lecture, I have tried to give you a brief overview of various practical applications of metal ions in diagnosis and therapeutic applications. It is clear that inorganic chemistry will have an important role to play in medicines in the future. In my lecture today, I will discuss more details about the platinum based anti-cancer drugs and their mechanistic actions. So, discovery of this cisplatin is an historic achievement that has had a worldwide impact on the lives of millions of cancer patients. The discovery stemmed directly from the pioneering work of Dr. Rosenberg, a physicist, and Lorette van Kamp, biologist, while studying the effects of an electric field of dividing E. coli bacteria in the biophysics department of Michigan State University in the early 1960s. Dr. Rosenberg's picture is shown over here. He is actually indeed sitting in front of picture of Albert Einstein. And here there is another picture is being shown that Dr. Rosenberg with Lorette van Kamp in the animal room at Michigan State University. This is what is Dr. Rosenberg. Now, cisplatin is indeed a therapeutic agent. Cisplatin is a chemotherapy drug used as therapy against testicular, most effective, ovarian, head, neck, bladder, cervical lymphomas, and cisplatin structure is shown over here. Now, although cisplatin is a tetra-coordinated transition metal, it has square planar geometry, platinum 2 D8 system. So, it stabilize square planar geometry, and there can be two possible structure, cis and trans. You see that cisplatin where this 2Cl is present in the cis position, and transplatin you see the 2Cl is in anti position. So, this is cisplatin, which is the most effective anti-cancer drug. Transplatin, however, is completely inactive. We will be talking about transplatin also, that why it is not active. Now, synthesis of cisplatin is shown over here. You can see that K2PtCl4, we start from that, and once you add potassium iodide, this Cl will be replaced by iodide, and then you replace one iodide with one molecule of ammonia. Then addition of another molecule of ammonia will replace another iodide, but they are in a cis position. Now, addition of silver nitrate precipitates AgI, and this both iodide would be replaced by water molecule as shown over here, and potassium chloride will indeed replace water to dichloro, and which are indeed the cisplatin, which is in platinum is the platinum 2 plus state. Now, you all know this DNA base pairing, and you see that this is thymine, adenine, guanine, cytosine and guanine. So, this kind of base pairing is there, and this is a double helix structure as shown over here. You see all details, the distances also given in this particular slide for your information. Now, cisplatin indeed able to bind this guanine nitrogen, this is actually N7 position. This it binds over here, and we will see soon the details of this kind of binding, but it binds in the N7 position of the guanine. This is the most preferred positions in DNA. Cisplatin has biochemical properties that allow it to produce interstrand and intrastrand cross linking in DNA. Both you see that intrastrand cross linking and interstrand cross linking also possible, as you can see over here, and there also can be possible this monoadduct formation and DNA protein cross link also a possible conformation. There is a possibility, and although this intrastrand cross link product is the major product, and we will talk about this in details. Now, most prevalent form of the 1 to interstrand cross linking, the DNA is indeed unwounded and bent towards the major group. Now, this is what is interstrand cross linking of the cisplatin with the DNA double helix. As you can see that platinum is coordinated with 2 nitrogen from 2 sides, and this is the intrastrand cross linking of the cisplatin with the DNA. DNA protein cross link is shown over here. You can see that this is DNA is coordinated with platinum in a monodentate fashion. This nitrogen is coordinated, and so DNA protein cross link is shown here, and bifunctional binding to guanine is also shown here, but these are all very minor possibilities, binding of cisplatin to DNA. So, this excess structure of major adduct of anticancer drug cisplatin is shown here. The cisplatin, as I have said earlier, the cisplatin preferentially bind this guanine nitrogen. This is the N7 position, and Lippard's group has made a ligand which is guanine deoxyribose phosphate dinucleotide. This is what is this ligand, and where you see that 2 guanine is over here, this is the ribose sugar which are linked with the phosphate, but this is a discrete molecule and small molecule, and once platinum binds to N7 of guanine is also clearly visible over here. Although this is not the DNA, but this is a part of the DNA they could able to make in the laboratory, and they could show the world that how indeed cisplatin can bind to the DNA. So, this is the X-ray structure of the molecule is displayed, where you can see that platinum is of course square planar, and this A2NH3 is coordinated here, and the guanidine nitrogen is coordinated in the other 2 sides as shown in the X structure. Now, indeed X structure of cisplatin with DNA is also been published. You see that this platinum is how it binds this guanine nitrogen N7 position, and this is once I have zoomed that portion over here, you can see very clearly how this platinum is actually coordinated to this guanidine nitrogen N7 position very clearly, and this has been also published long back by Lippard group in 1995. Now, let us talk about the mechanistic action of cisplatin, that how indeed cisplatin works. And as you can see that this is what is the cisplatin, and then this is the plasma membrane. By diffusion and by other means also cisplatin enters within the cell. Then they hydrolyze the chloride stepwise to form diaqueous species. And why cisplatin do not hydrolyze in the blood? Because it has been found that this chloride concentration is much more 104 millimolar solution of chloride there in the blood. However, within this cell chloride concentration is only 4 to 10 millimole in cytoplasm, and that indeed helps cisplatin to hydrolyze, and as you can see that this is 2 water molecule is replacing this chloride, and then this cytosine and guanine DNA base pair is shown over here, and you can see that cisplatin binds very clearly the interstant adduct, which is actually the major product, and this is shown over here, and once cisplatin binds to the DNA, then it goes to the cellular processing of the DNA lesions leading to apoptosis. So, cisplatin comes inside the cell, it binds with the DNA and forms the interstant product, and then cell becomes dead, and this is what is happening with cisplatin. Now, cisplatin, as I have said, is a therapeutic agent. This is the structure of cisplatin, where 2Cl is in a cis position, and transplatin, this is where 2Cl is in trans position. They are structurally very similar. However, cisplatin is active anticancer agent, whereas transplatin is not active. The first difference in the anticancer activity of cisplatin and transplatin may seem very surprising at first, giving their structural similarity. However, this fact has been rationalized in terms of the greater reactivity of transplatin compared to cisplatin. Transplatin acquires approximately 4 times faster than cisplatin, and following a 4-hour incubation with red blood cells, transplatin reacts with 70% of the glutathione, whereas cisplatin reacts with only 35%. This greater reactivity can be rationalized in terms of trans effect. The high reactivity of transplatin results in rapid deactivation of the complex owing to site reactions on the way to its target that are likely to contribute to its lack of anticancer activity, and that is the reason transplatin is not indeed active. The major limitations to cisplatin therapy is its side effect. It has severe side effects. For example, the common side effects are nausea, vomiting and kidney damage, and slightly less common side effects are hearing loss and numbness or tingling in hands and feet, temporary taste alteration, loss of appetite, diarrhea or allergic reaction. Sometimes people lose hair also. Now, what is the reason of this toxicity? The cellular environment is indeed rich in thiol containing species that can bind to cisplatin and immediately deactivate it. This deactivation is due to strong coordination of the soft sulfur donor coordinating to soft platinum 2 in preference to harder ligands such as amine nitrogen donors, a concept which all of you know as hard soft acid base SAP principle. Now, in order to minimize those toxicity, lots of people trying to make molecules, and there are certain structural guidelines one should have before they design this platinum based drug. The compound should have 2 amine group with a cis geometry, as those with trans geometry are completely inactive. It is necessary, although not sufficient, for the compound to have 2 leaving groups that are cis with respect to one another. I mean, leaving groups are those groups on the molecule that are most easily lost. The ease with which the leaving groups are able to be lost affect the activity and the toxicity of the compound. It is preferable for the leaving groups to be only moderately easy to remove. It only be removed inside the cell. If it be removed outside the cell, then it creates lot of toxicity, and also the compound should be neutral. Okay. So, these are the criteria one should follow before it design some new anti-cancer drug. Let us look back cisplatin once again. Cisplatin was indeed approved for use in 1978, and cisplatin is responsible for the cure of over 90% of testicular cancer cases, and it plays a vital role in the treatment of cancers such as ovarian, head and neck cancer, bladder cancer, cervical cancer, melanoma as well as several others, regardless of many side effects in patients while using cisplatin. It is one of the most prescribed anti-cancer drug till today by the doctor. The problem with cisplatin is it has severe side effect, toxicity to kidney and nervous system, and also its resistance. Carboplatin also being designed, which are now used also in clinically and which are less toxic and much lesser side effects. So, carboplatin is now very popular. It can also replace cisplatin because it is less toxic. Now, bidentate ligands is more stable, slower reaction in the body, and some of this anti-cancer drug is also shown over here. You see that oxaliplatin, which is used for treatment of colon cancer, and then another platinum complex which actually overcome the resistance also. So, clinically approved and marketed platinum anti-cancer drugs are displayed over here. As one can see, there are several such platinum 2 complex like cisplatin, carboplatin, oxaliplatin, nedaplatin, heptaplatin and lobaplatin is shown here, and this is what is the list where you can see that what is the research name of this molecule and what is indeed the trade name and when it has been approved and where it is being utilized. As one can see that cisplatin, carboplatin, oxaliplatin are utilized globally, whereas nedaplatin is in use in Japan, which has been approval granted in 1995. Then heptaplatin, which are now used in Korea and approved in 1999, and lobaplatin, which are approved in 2010, has been in use in China now. The problem is all this platinum based anti-cancer drug has severe side effect, and in order to reduce that, people have been trying hard to make different molecules and try to think that how one can reduce the toxicity of this platinum based anti-cancer drug. One such attempt was like if one can make platinum 4 product. Two non-living groups are here, platinum is in octahedral structure and these 2 X is the living group, and there are 2R group, which one can play with that. Now, this anticancer potential of platinum 4 agent has been recognized from the time of the original discovery of the biological properties of cisplatin, but their clinical value has only more recently been realized. The physicochemical properties of platinum 4 agents differ significantly from those of their platinum 2 counterparts. It is generally accepted that inert platinum 4 complexes are prodrugs that must be reduced to their more reactive platinum 2 analogues to exert anticancer activity, as the platinum 2 analog is the active species. This is the diagram where you can see that there is a scope to vary different groups, and you can improve your anticancer drug, and you can play in such a way that you can make your drug more friendly, less toxic. That would be of great help for the community. Unlike square planar platinum 2 complexes, platinum 4 complexes are nearly always 6 coordinate and adopt octahedral geometries. The standard kinetically much more inert coordination sphere of platinum 4 is more resistant to ligand substitution reaction than 4 coordinate ligand to center, thus minimizing unwanted site reaction with biomolecules prior to DNA binding. Moreover, the 2 extra ligand afforded by low spin D6 platinum 4 center provide a means to impact and fine tune desired biological properties such as lipophilicity, redox stability, cancer cell targeting, and improved cellular uptake. If one can possibly use platinum 4 prodrug, and by that way one can reduce the toxicity, and indeed there are several such platinum 4 agents that are undergoing clinical trials at present, and you can see some of these structures are shown here. However, as I have just said, the platinum 4 agents are actually prodrug. The real drug would be platinum 2 plus and square planar. So, what is happening inside the cell? This undergo a reduction to produce this square planar platinum 2 complex, which is actually the active anticancer agent in our body. So, where we are going now? Lots of new drug and new possibilities for metals in medicines. There is much research to be done. We need to find anticancer drug with less toxicity, less side effect, and then it would be actually very useful for its applications. In summary, I have discussed today more details about platinum based anticancer drugs and their mechanistic action. Hope you enjoy the chemistry I have shown in last several weeks, and thank you very much.